All right, we're going to get started. Thank you so much, everyone, for coming back. And thank you to the Koch Institute for throwing a great event so far. I'm Rebecca Spalding. I'm a biotech journalist at Bloomberg, where I cover the business of drugs and the future of healthcare. So this panel is, we have a wealth of panelists, as you all know. I'm going to introduce them shortly. I'll try to keep it brief, but they all have many accolades. But you know, one of the things that we think about a lot, not just at Bloomberg, but also in the public, is taking these exciting insights, this exciting research, and actually translating it to therapeutics for patients. So I think it's a great topic for a panel. So to my left is John Maraganore. He's the Chief Executive Officer of Alnylam, which is expecting its first FDA approval this year for patisiran, which would be the first RNAI therapeutic available. Prior to Alnylam, Dr. Maraganore served as the officer and member of the management team for Millennium Pharmaceuticals, and has also held senior positions at Biogen. He's the current chair of Bio, and has been a vocal advocate for the industry during some tumultuous times.
Including this week.
Paula Hammond is the David H. Koch Professor in Engineering and the head of the Department of Chemical Engineering at MIT. She's a member of the Marvel Center of Cancer Nanomedicine at the Koch Institute, the MIT Energy Initiative, and the founding member of the MIT Institute for Soldier Nanotechnology. She's a recipient of many awards and has been elected to the National Academy of Engineering, the National Academy of Medicine, the American Academy of Arts and Sciences. She's also the director and co-founder of Layer Bio, a drug delivery startup based on particle technology developed in her lab at MIT. Noubar Afeyan is the founder and CEO of Flagship Pioneering. During his long career as an inventor, entrepreneur, CEO, and venture capitalist, he has co-founded and helped build over 35 successful life science and technology startups. He serves on a number of public and private company boards, including Kodiak Biosciences, Kaleido Biosciences, and Moderna Therapeutics. At MIT, he's deeply involved with the Sloan School of Management, where he's taught courses on technology entrepreneurship since 2000. He is a member of the corporation, otherwise known as the Board of Trustees of MIT. Bob Langer is the David H. Koch Institute Professor at MIT. He's the most cited engineer in history, and his patents have been licensed or sub-licensed to over 250 pharmaceutical, chemical, biotechnology, and medical device companies. He's the recipient of many awards, and is also one of the very few individuals ever elected to the Institute of Medicine and the National Academy of Sciences, the National Academy of Engineering, and the National Academy of Inventors. He is a member of the Marble Center for Cancer Nanomedicine at the Koch Institute and the Koch Institute, and he has served as a member on the FDA's Science Board, the FDA's highest advisory board. Christian Rieken is the founder and chief scientific officer of Crystal Therapeutics, which develops the next generation of targeted nanomedicines to treat various diseases, in particular cancer. She is the recipient of several prizes and awards, including the Simon Steven Gazelle Award in 2008 and the Knowledge and Growth Inspiring Young Scientist Award in 2000. She is a pharmacist by training and holds a PhD in pharmaceutics from Utrecht University. And lastly, Michelle Bradbury is the co-director of the Memorial Sloan Kettering Cornell Center for Translation of Cancer Nanomedicines. She is a board-certified diagnostic radiologist and the director of Intraoperative Imaging Program at Memorial Sloan Kettering. She earned her PhD in radiological sciences at MIT. She serves as the principal investigator of multiple nanoparticle-based clinical trials and is also a member of various scientific advisory boards. She is also the founding member of Lucida Oncology, a startup focused on particle-based clinical trials. Ladies and gentlemen, our panelists, thank you for being here.
Any questions?
So Bob, let's start with you. You're obviously a pioneer in polymer science and drug delivery. How would you describe to a member of the general public who may be here about the benefits of using nanoscale approaches in oncology in particular?
Yeah. Well, I think we've heard about some of those already this morning, but I mean, to me, some of the advantages are that you can deliver drugs better, you can target them to tumors perhaps better. You can also, as I'm sure you'll hear from John and Noubar, maybe me, use nanoparticles to deliver molecules like siRNA and mRNA, new genetic medicines that I think will transform medicine. I think in addition, you can cross different barriers, like Mark Davis talked about crossing the blood-brain barrier, and people have looked at them for crossing other barriers as well. And then I think there's a whole area of diagnostics because of the enormous surface area of particles and other properties. There's new types of diagnostics. I imagine Sangeeta will talk about that. In fact, Noubar and I and some of the other professors at MIT, like Tyler Jacks and Mike Sema, we started a company, T2 Biosystems, which does ultra-rapid diagnostics and was able to take a diagnostic, say, this isn't, for example, blood testing, but enabled you to move it from 5 hours, where a lot of people would die, to 3 hours, 5 days, where a lot of people would die, to just 3 hours. And that just got approved by the FDA a couple of weeks ago. But so I think there's just so many things that are transforming medicine that use nanotechnology. And I hope, given what you're hearing both this morning and this afternoon, there'll be a lot more.
I'm curious kind of how you've seen the field mature since you first started, since nanotech first started to become something that a lot of people were becoming interested in. How have you seen Cambridge change?
At Cambridge, well, first I'd say nanoparticles have had, and as any technology, there's a lot of ups and downs. As Mark Davis said to me, it's hard. But Cambridge, I mean, I can tell you, and it's not just nanotechnology. When I was a graduate student in MIT in the 1970s, the area around MIT was a slum. I mean, it was just, there were no hotels. The only restaurant around was a not very good submarine shop. And mostly there were these bars like Father's 2 and 3. And what's happened over 40 years in Cambridge because of the biotech revolution is now it has the highest concentration of biotechnology companies in the world. And not only that, like I remember I had to testify in front of the Cambridge City Council a couple of years ago, and they were asking me about this, and I mentioned the point about restaurants, and I happened to look in the Boston Globe the day before, and they listed the 50 best restaurants in Boston, and 7 of them were within 4 blocks of my office. So it's, and there's lots of hotels now. So it's changed. I don't think there's been a transformation like this anywhere that I've seen.
And, John, you've also been there to see the transformation. Alnylam's overnight success that began in 2002. I'm curious, when you guys were first starting in 2002, 2003, what was the thinking then about nano and using something like RNAi to actually make a therapeutic?
Well, just yesterday we celebrated our Sweet 16 birthday, so it has been an overnight success. Not really. Obviously, it takes enormous amounts of time. But from the beginning, we always knew that delivery of small interfering RNA was going to be the major challenge to build a whole new class of medicines, and we knew that we had to look at a whole range of different approaches to ultimately get these molecules to be safely and effectively delivered into cells where they needed to operate to mediate the intended effect. And so, nanoparticle approaches, lipid-based nanoparticle approaches, and even non-lipid-based nanoparticle approaches were always in our crosshairs of a type of technology that could be deployed, in principle, to do what we wanted to do. And it's really gratifying that we're now literally weeks away from the first approval of an RNAi therapeutic that uses a nanoparticle approach, a lipid-based nanoparticle approach that was pioneered, almost through a Manhattan Project-like effort in the company between 2002 and roughly 2010, 2011, where working with, internally, working with other companies, working with some great academic groups, including Bob's and Mark Davis's and others, we really figured out how to ultimately make nanoparticle technology that allows us to deliver RNAi therapeutics.
And so it sounds like that was very much one of the big challenges that you guys faced, that after you figured out the therapeutic side of things, it was, well, how do we actually make this into a drug? How do we deliver it to patients?
Absolutely. Yeah, it really was. If you look at just the enormity of the translational effort, not only did it take us 16 years to get to the stage, which of course is not a short period of time, but it's taken us about $2.5 billion of invested capital. And we've raised over $4 billion as a company to date. So just the scale of what it takes to do that is profoundly large. And it takes a lot of very, very perseverant efforts, because we've had our near-death moments many times. Many times people thought we were done and it was over. And we stuck with it. We stuck with it because the science told us to stick with it. But we've been fortunate that we've been able to get over that delivery hurdle and now really build important medicines.
And Paula, I know, in your other life, you're the co-founder of Layer Bio. I'm hoping you could talk a little bit more about that, but also it seems that you're past that proof of concept phase where you do believe you have something that could be potentially helpful to patients, certainly something that you want to study further. When you hear a number like $2.5 billion, I mean, how are you taking your insight from the lab and actually making sure it can hopefully reach patients?
Yes, I think it's important not to be scared by the numbers because although they're very real, everything starts with smaller efforts and proof of concept, and then as you begin to move forward, you find whatever you can to leverage your technology to the next level. Layer Bio is a company that uses a very simple nanolayering technique to incorporate biologics and nucleic acids onto a range of different scaffolds for regenerative medicine. And we think that there are some possibilities in cancer as well. I think that to leverage this kind of technology, you have to be agile and you need partners who will work with you. So for me, it's been a matter of finding members of lab or members of my collaborative team who are willing to move things forward. Sometimes you can start with the simplest, lowest hanging fruit and begin to look at where you can get funds to launch or move that a step forward, finding out what's required to get an IND and get approval, and finding out which partners might be available. It's also important to remember that there's more than one way to translate a technology. And it may end up that you need to start your own company, but you can also perhaps find very meaningful partnerships with larger companies. And in some cases, of course, you do both. And your smaller company begins to work closely. Layer Bio works closely with larger companies to look at translational applications.
And Michelle, I know that you have also gone through this process at Merlin Kettering. I was hoping you could tell the audience a little more about your platform, but also some of the challenges and also how you overcame those challenges in translating your technology.
Yeah, so in the technology we're using at Memorial Sloan Kettering, that platform, the particle itself, is an ultra-small silicon nanoparticle that was developed at Cornell University, Ithaca, by my collaborator, and we took this from bench to bedside over many, many years. I mean, I think the issues were that you have a highly engineered, very small particle that's 6 nanometers in diameter, so the size of albumin. And you want to get the surface chemistry just right. You know, it's a hospital setting, and so the safety issue of this was for such a particle that, has a dye encapsulated within it for fluorescence imaging. We're using this continuously in the operating room, in the clinic. And so we also had targeting moieties and a PET label on the surface. We've put, drug linkers on. So the question is, how do you really optimize the surface chemistry properly for the given indication in a manner where it's stable and reproducible and it's a safe product? And also the dosing of such a platform in a patient. What should that dose be? So we use very tiny doses when we started out for a first-in-human trial, a microdose. So there were a lot of challenges, I think, because there were no, at the time we were doing this, having a targeted dual modality platform in the clinic for such an indication as a nanoparticle, didn't exist. And there were no real FDA guidances around such highly engineered products. And so I think the mature infrastructure of the hospital setting has helped evolve these types of products for clinical care. But it took a long time to get this right and to understand the stoichiometry and what's going to be optimal for a given indication surgically and even for oncologic applications. So I would say that the scientific component of this was enormous. I mean, the science took a very long time, and there's no point doing a trial if the science hasn't been worked out well for such a platform. So I think the trials now I'm in a phase 2 trial in the operating room trying to map nodal disease for various cancers. And it's done as a stage development for the surgeon because the surgeon wants to see the disease before treating the disease. So that's a critical component to us moving the technology forward in the clinic. So we've evolved some therapeutic options. I think another area that's really important for the field too is the fact that particles have unusual properties, many of them. So there are so-called self-therapeutic properties where even the technologies themselves can be therapeutic without a drug. So there's some key examples of this, and it's very exciting to see more and more of these sorts of examples out there. I mean, for our particle, we found a cell death program, just raising the concentration of the particle under metabolically deprived conditions. There's a cell death program for apoptosis, which is iron-driven, ROS-driven, and can kill cancer cells in a synchronous way. And I think that we can find interesting combination therapeutics utilizing such programmed cell death mechanisms. So it's very exciting. I think a number of us are seeing these types of effects, but for us, it's been a very long haul to get this into the clinic and where we are now.
And now, some of the most exciting companies in Cambridge grew out of Flagship. Noubar, when we were speaking earlier this week, you mentioned that Flagship's process is almost the reverse of an academic process. Can you walk us through Flagship's process and how some of these companies, Moderna, plenty of others, have come out of that?
Well, I think it's in the context of translation that you were asking. And so I think you can, if you have, as we just heard, a well-developed piece of science that can be applied and over time is ready for translation, then you can basically apply yourself to translation and find applications and put things in the clinic. Often the things that we end up working on, the science is largely enabling. It's early, but there's really no sense of where applications may be found. And we find that staring at the science isn't necessarily the best way to figure out where it might end up 5, 10, 15 years later. And that rather staring at where it might end up for a while and figuring out of all the possible things you could imagine doing, which ones might both tie to value and that is solve a real problem and can be connected to science that exists today is just the opposite way to go. And so we often nowadays look at science kind of looking backwards from the future and say which science combined in which ways might enable us to get there. And it seems abstract now, but what I was saying is there's kind of almost reverse translation you can think about, which is that you can envision what a drug needs to be able to do and come back and say, do I want to do this with a small molecule or a protein particle combination or a cell or a nucleated cell? And from that point of view, it's less about the component technology and more about the solution you're trying to drive. And I think, and from that point of view, since we're here talking about nanomedicine, as I was thinking coming into this, Flagship's been around as long as nanomedicine has been talked about, or certainly nanotechnology, which is 99, 2000, if you look at the publication rates, that's around when it started taking off. And I think in our existence, we've been involved in at least nine different companies over the last 17 years that have in one way or the other put either nanoparticles in therapeutic or diagnostic use. And there's just been a massive amount of learning over time, a lot of it coming out of the work at MIT and surrounding startups, but other places too. And so I think we're now in a place where it's becoming part of the reliable arsenal of tools available to dare to do things that in the past used to seem completely speculative. I think we're getting more and more confident. We haven't yet tackled specificity issues. We haven't yet tackled a number of other things that we'd like to be able to do, but this has come a very long way, and I consider it an integral part of designing kind of functional drugs of the future.
And Christian, you're the Chief Scientific Officer of Crystal Therapeutics. I'm curious if you could describe your development approach there and how it's evolved as you're translating nanomedicine to patients.
Right. Yeah, so maybe as a reflection, back in 2007, I defended my PhD thesis, which were on polymeric micelles back in the Netherlands, by the way, And then, of course, quite some nanomedicines have already been developed, but I also saw already back then, I had one patent and I had some differentiation versus these systems already developed. So I thought, well, maybe let's give it a go without knowing, of course, that I would be sitting here 11 years later and having a phase 2 study being initiated in only a couple of weeks. So meaning that along the way, like in the last 11 years, we've made some major progress in terms of scientific development, meaning that we build on all the developments in the worldwide development of nanomedicines. We incorporated in our own product platform development. But at the same time, it also holds true that we learn from the setbacks of other companies, meaning that we try to follow more or less a science-based approach, that we really want to tick all the boxes, and particularly the biological barriers that we've heard about. And also in the clinical evaluation, we want to make sure that we actually going to bring the nanoparticle to the tumor tissue. So rather than the more classical approach of how to say it, administer drug and measure only efficacy, what we're doing is two phase 1 studies in parallel to actually take a look at a clinical side whether the nanoparticle is reaching the tumor tissue. And that's really the attribute to all the developments ongoing in the past. So that's what we really have to learn from a worldwide perspective. So not only here in Cambridge, but also back in Europe. And maybe to reflect on all the developments, I heard some values about investments. Unfortunately, the investment climate in Europe is not as big as what you mentioned. So, but still, there is quite some investment in these type of innovative approaches, as long as you have some supportive mechanism of action studies demonstrating that your claims are also going to give ultimately an added value for the patients, and thus rewarding the product. So yes, I'm very pleased to be capable of having this progress so far and learning from what all has been done in the field.
I'm curious, Bob, you've obviously had a long track record of taking insights from the lab to commercialization in various ways. How do you know when something's ready, when it's past that proof of concept phase, if you will, and is ready for commercialization? Or at least to begin that path, let's say.
Well, I don't know that you're ever 100% sure, but I mean, to me, I guess the kinds of criteria I've used, and usually people have worked on these things in the lab for 4 or 5 years, like somebody's thesis or somebody's postdoctoral project, and hopefully by that time they've got a paper. And again, for companies that you really want to see funded, hopefully the person's made some type of breakthrough, ideally published it in a journal like Science or Nature, or maybe even have a few papers, that they also have really good intellectual property, that we filed. That probably it's some type of platform technology, which, like a lot of the companies that, all the companies, all the things you've heard about today are platform technologies, because they can be used for many, many different things, not just cancer, but all kinds of cancer and all kinds of other diseases. And usually for me, the other criteria that I've often had is, in a lot of the cases, that the people in the lab who've done it, they really want to start a company. I mean, that's been their passion. They've worked on a thing for 4 or 5 years, and they want to see work that they've done or work related to what they've done help people. And so starting a company is a great, there's no better way to do it than that.
Does anyone else have any?
I think, Noubar started a lot of companies, so he'd probably have some really good insights. But I mean, I think the point that Bob made on the people side is really important. If you have a team, whether it's the scientists in the lab that are the scientists that's doing the work, or other people around that really have the passion for something, that is huge. That is huge, and that goes a lot to feel comfortable about it being ready to go into a translational mode, for sure. You need that. That's a fundamental thing you need. I mean, Noubar, you should comment as well.
Yeah, I think that there's a couple of types of companies in the biotech industry. One takes things that are close to being drugs and then tries them on people and then creates value. It's a bit of a lottery. If it works, great. If it doesn't, it doesn't. And I think we're not talking about those kinds of companies. The other kind of which is a perfectly good thing to do, but just not, I don't think it's going to lend itself to nanotechnology anytime soon. I think the other kind of companies, the ones we're talking about, are really kind of fairly long journeys, in the sense that you're not actually, as Bob said, thinking of one drug, but you're thinking of many, and you're trying to figure out what does the category involve and what do I need to think about. And so there's a lot of learnings so the company tends to be founded, over and over and over and over again. I mean, this notion of founding a company, which we all consider kind of the, when we print out business cards and name something, it's kind of a curious notion when you're developing a platform because, I don't, as John said, at a minimum, every time a company is near death, it's founded again, meaning that, you survive and you have a second chance. And so I think it's important to understand that. And so detecting when something is ripe, for me, has never been an interesting question. I think that I actually would rather pick something that's really, really not ripe and figure out how to ripen it, divide it, multiply it, decorate it, do all sorts of other things, because that's where the value comes from. It never ends up being about just picking the ripe thing and having been right in your picking. I just don't, that's why I think most of the people in the investment side of this business are not investors at all. They're basically scientists who are professionally working with entrepreneurs to help companies come to life. That's a long journey. It's not about picking.
I mean, we were, just to jump in, I mean, we were founded in 2002, and, Fire and Mello published their seminal paper in Nature in '98, and then it was viewed as not being relevant in mammalian systems, and then it really was the work of Tom Tuschl and the paper that he published in '01 that sort of drove the reality that, oh, this could be interesting. And so we were really started only about a year after that Tuschl paper. And there was a previous paper in 2000 from Zamor and others, but that was relatively young science, right? But it was impactful, and it was worthy of investment to figure out if you can make it work.
Let me just add one other thing, Rebecca, on this, because just for people who are thinking of starting these companies, I think each of us have examples of this. There's, we as scientists, engineers, we're trained with understanding facts, analyzing, modeling, predicting, doing all sorts of things with facts. But in this kind of an effort, there's only that much you can analyze, there's only that much you can quantify, and the rest is a leap of faith. And so what you would be asking is, when is something good enough for people to make a leap of faith? And the reason I think Bob's saying you got to look at the people is that at the end, it's a human endeavor to say, if I only know as much as I'm going to know, and the rest I'm going to make real. And so it's the judgment is, when is it right to make a leap of faith? It's completely, I don't know of any great company that rationally could have been proven to be the right time to start. A lot of this is just leap of faith.
Paul, I was hoping that you could weigh in, and you as well, Michelle. We were talking earlier this week that plenty of brilliant people who, you have a company, you also have another big job. There's, I'm sure, plenty of brilliant people in the audience who also might have multiple roles. So how do you think about going and founding a company in the context of academia, a research hospital, that sort of thing?
I think it depends on what role you already have. And as an investigator or a PI at a major institute and also a faculty member, I have one primary role, which is to create an environment in which all of these wonderful ideas can grow and develop. I also want to see them translated, but that requires a partnership on some level. For me, it is important to have that additional person who wants to move the technology forward. And Noubar and Bob put it extremely well, that if there is a member of the team who is excited and really believes in that principle, then it will happen. Because quite frankly, we have incredibly able, capable people working on these problems, and if they see the potential, then it becomes real. And I have seen that not only in my own lab, but as department head in the labs of many of my colleagues. So for me, the idea is to create the platform and to make it possible, and to partner with those in my team who can actually translate it. The other option is, of course, to begin to work with other partners and find other paths toward translating the technology. But because of the definition of my own role, I will never be able to become the CTO or CEO of a company. By definition, I think that the answer then varies depending on what your perspective is. But for the young people in the audience who are at the very beginning of their career, it's a great opportunity to grow and become someone who has learned not only about how to implement your own ideas and make them real, but also how to manage people, how to work with different institutions, how to convince others. I think it's an incredible opportunity in that sense.
Michelle, could you weigh in as well? That I know that you've had multiple roles as you've been building this.
Yeah, I agree with what Paula said. Your partners, you know, in the hospital, of course, other clinicians, in this case, let's say surgeons, who really see that there's an unmet need. We just don't have the tools to do the right kind of job in the operating room that we would like to do. You know, they are partners for a very long time, and it's a sustained effort. You know, it's not a year or two. They realize over time, wow, we've been working together almost a decade. And, you know, you really can develop something quite well with the right kind of partners internally. So you wear a hat between, you know, the initial development of the platform, moving into a clinical type role where you make sure that the design of the trials are addressing the unmet needs. You know, what are our endpoints? What are the best endpoints for this? Sometimes there are, you know, it's an untapped area. You have to really create something that the FDA would look at and say, this looks like a robust design. And is it reproducible? You know, can the surgeon always pick it up and it shows the same effect? And then finally, you get to some stage where that academic development, you know, you really, I think partnering with pharma and other individuals and other entities can really lift the nature of how you would extend it to the more advanced phase trials. So at each stage of development, you're bringing in new partners to help with that. And I think many of us can naturally expand into these different roles because we're evolving with the technology to do that.
I think it is important, as Michelle said, to really leverage those partnerships and to remember you don't have to be everything for your starting entity. Recognize that there's a huge amount of expertise near you, and I think we have many microcosms of people who can give advice or perspective. Working with an industrial partner gives you an idea of what is really needed or what may be valid. Working with someone who is on the board of other companies can give you a perspective on how a company can be well structured to enable innovation. So using our partnerships and relationships the best that we can actually greatly enable a company. And the nice thing about early stages of companies is that roles can change and they don't have to be very highly defined.
And I know clinical trial design came up in an earlier talk, Michelle. I know that we've talked about it as well. And you know, I'm just curious, going back to John, to your point, that when you started, it was very, very early science. Now you're looking at, hopefully, a drug approval this summer. So what were sort of the learnings from actually taking science that the FDA, the industry, had never really seen before, and not only proving, you know, RNAi to them, but also making sure that you were designing your clinical trials in the right way and thinking about that?
Yeah, I mean, there's just so much to I mean, when you have a whole new technology approach, and I'm seeing the same thing happening now with gene editing. I'm seeing some of this learning, you know, happen in other technologies too. I mean, there are just steps in the road that you can't predict that you need to essentially generate the evidence around, whether it's evidence around safety or efficacy, but you need to generate the evidence and do it in a high quality way. And that's true in your engagement with the scientific community. For example, we took a very explicit decision as a company when we got started to publish our science and use the peer review process of reviewing science and publishing scientific results as the right way to communicate the progress that we were making. And you know, we did that knowing that we would enable our competitors to also learn from our science and potentially compete with us. But you know, frankly, we did the value trade-off and said, you know, the benefit of communicating our science in a high quality, high integrity type and very transparent manner greatly outweighed the risk of, you know, mysticism or, you know, uncertainty or, you know, disbelief that can happen on the other side. And you know, it's interesting. You know, obviously, you know, I'm just picked up Bad Blood to read that and the Theranos story and so forth. And, you know, it is interesting how if you don't create the right transparency around a technology, around a platform, around a way of advancing a new approach, you're just going to be, unfortunately, potentially in the world of disbelief. And you're better off, enabling competitors and being believed and credible than you are being disbelieved at the end of the day. So it was an explicit decision on our part. And we still to this day celebrate when we publish papers. If you come into our offices, we have these big posters of the front pages of our different papers that we publish over the years. And we think that's really important about how we do it. So I think to answer your question, you just have to use solid science and communication of the science as a way of generating the needed evidence so that the uncertainties around something that's new can basically be appropriately weighed in the context of a new medicine where you have to measure the risk and the benefit of a new technology from an approval perspective.
Absolutely. And Bob, I'm curious how you've approached some of those conversations over your time, bringing brand new things to commercialization.
Well, I think what John said is right. We if you're an academic, we publish everything.
Sure.
We patent everything too. But I think that we are. To me, I want to make sure that the students get good papers and get good jobs, and so I think what we've tried to do is, really encourage in our lab a spirit of invention, a spirit of discovery, and when things get to a certain point and students want to take those discoveries, and if they've really crossed some of those bars that I mentioned, hurdles that I mentioned earlier in terms of publication and patents and in vivo feasibility, then very often we have worked with venture capitalists like Noubar and others to start companies, and so that's just been a wonderful model for the people in the laboratory.
I'm curious, now that nano, it's been around since 1999, 2000, when people first started talking about. I'm curious if I can get all your thoughts on where you really see the field heading in the next 10, 20 years, particularly as it relates, of course, to cancer, but more generally how it might converge with other areas that are currently in labs and pipelines around the country and the world. Michelle, do you want to start?
So we're at such an exciting time. With so many fields that are converging on what we're doing. And it feels like we are reinventing ourselves all the time. And many of the themes talked about this morning, where we're going, the doors are opening with lots of different areas that we can work with. For example, I'm a radiologist, and so I look at machine learning, and that was presented this morning, and how we can really tap into that area to really help us do a better job with selecting patients, let's say, stratifying patients for our trials. What kinds of features do we want to select from our images to help us with this? So, these deep learning algorithms, so I see there's a lot of focus now on just having more precision medicine inserted into the trials. I think that's starting to grow. Another area, augmented reality. So I'm in the operating room with surgeons, and how can we facilitate the surgical approach in our patients? How can we help surgeons do a better job, let's say, or make it a bit, facilitate the operation in ways that they couldn't really think even about before, because all they have are scans done before the operation. And also I think that in another area is really, we've come to understand some of the limitations of some of the prior developments in nano over the last 30 some odd years. We see where some of the difficulties are. We've worked with these designs, and now maybe we can tap a number of different therapies that have been sitting on the shelf, or we could work with those in a better way now and advance some of these alongside the newer generation drugs, perhaps to do a better job with our sustaining the effect in patients. So those are some of the areas that I think, from my perspective, are really vital to really pushing the frontiers of where we are today.
Christian?
Yeah, if I can add to that, in terms of, I'm a pharmacist, so what we do is we develop nanomedicines as therapeutic agents. I think the field is moving towards more theragnostic approaches, so to make sure that as we get more and more insight that this EPR effect is so heterogeneous in patients, to make sure that we have really appropriate tools to select only those patients that will actually benefit of it. And I think it's a combination of what the former speaker already said about understanding what's happening, so the imaging tools, but also having more and more tools in hands to modify the nanoparticle with all kind of chemical, pharmaceutical properties, combining it with more and more detailed insight into biology, immuno-oncology, and getting into all these different aspects combined into novel treatments, which probably comprise more than one single disease pathway, and then actually measuring each time in the patient what's happening, and based on the results, go for more personalized precision approaches. I think that's going to take not 1 year, but probably 10 years or maybe 20 years, but that's the field we're heading towards.
Yeah, so I think in cancer, to me, one of the big exciting areas which I mentioned is really genetic medicine, the type of thing that Alnylam's doing at Moderna. Pick a cancer example, which Noubar might want to expand upon, is what we've done there is this whole idea of personalized cancer vaccines, which I think is a very exciting area, but you put them in nanoparticles to inject them into the patient, into the tumor. But I would say really more broadly, there's almost no area that I think nanotechnology won't reach. Just even thinking about different diseases in different parts of the body. For example, we started a company with one of my students called Kala, and the whole idea there, that's a public company now, and they're using nanoparticles. We're using them to treat inflammation after surgery, and I expect that to be approved in the next couple of months. It's also being used for dry eye and other things. We started a company where, again, Flagship and Polaris were founders, Selecta, and that's also a public company, and they're using nanoparticles to affect the immune system so that you might get tolerance better. I think there's really almost every part of the body. Mark talked about in his talk the brain. There's just so many different places where nanotechnology can go and is going.
Yeah, well, it's kind of hard to go last here, so I feel bad for you guys. Look, I think speculating about 15, 20 years from now, I think speculating about a year from now looks already pretty difficult to do. I think that one of the ultimate goals is going to be a programmable way to get anything into any cell, specifically. And I think that, and I say programmable way because if it's kind of happening haphazardly and every time you have to do it again, you start from scratch, it's the cost and inefficiency is dramatic, and luck is not a strategy. So sometimes people find things that get into a certain type of cell because it had some receptor by chance, and it's of no use to anything else because it just you used some biological phenomenon. So I think we will have. I think that the sophistication of the biological systems that we're trying to carefully alter is such that we're going to need with nanotechnology to almost replicate or instantiate that level of modularity and precision that already exists in biology. This is why a lot of times people use biology to do nanotechnology with medicine, because you have kind of such an exciting tool set there to work with. But if you're going to do it with materials so that you have immune advantages and other things, I think you just need to, that to me would be, if somebody came along and said, I have a way to take anything I put inside of a nanoparticle and deliver it to specific cells to the extent that I need to do it and not to others, I think all the rest would pale in comparison.
Excellent. I love all of these ideas. I think an additional area that nanomedicine will begin to appear in is in essentially lengthening or extending the quality of our lives. So aging is one of the big areas of interest right now. I don't think aging is something that we need to cure. It's not a disease. But I do think that we can use nanomedicine as a way to enhance and extend the function of our tissues in ways that will actually be meaningful. I think it can also be something that impacts those in countries where nanomedicine and other higher cost approaches have not been able to benefit by developing cheaper or low cost nanotechnologies that can improve everything from health monitoring to fitness to, I don't know if there'll ever be anything along the lines of nano nutrition, but I think that there are ways in which we can address a number of the issues around health, hunger, and other issues that impact humans on a broader scale.
Well, it is harder to be last, you know. I agree with everybody's comments. I'll make 2 points. One is, you know, this really is a remarkable time. Michelle said it earlier in terms of the innovation coming out of science and both in the academic setting and also in the company setting. Really remarkable time. And interestingly, the shape and flavor of our medicines are changing quite a bit. They're much more complex as modalities go. They're no longer sort of a few atoms strung together, you know, and small and flat. They're now very complex, and they go beyond recombinant DNA and monoclonals, which have been obviously so successful in medicine. And these more complicated systems and modalities, whether it's cell therapy or gene therapy or RNA therapy or whatever, will all benefit by nanotechnology. It's sort of an overlapping feature of how many of these approaches will be ultimately successful. And I think I can see in the future that, you know, there are many nanomedicine-based approaches that, you know, work together with gene approaches or RNA approaches, whether it's messenger RNA or gene editing approaches, and that's going to change the flavor of the type of medicine that we are used to today. That's point number one. Point number two is an area that I think is really interesting is how we can change the adherence to medicines. You know, one of the biggest problems is patients not taking their medicine and therefore not having the benefit of those medicines. And I think that an area where nanomedicines in particular can be very impactful is to change the adherence to medicines, the durability of a given medicine's effects in the body. There's been a lot of progress already from that dimension in what has gone on, but I think that could be another area that's very important.
I know you wear two hats as well. You're the chair of bio. So I'm curious how you're preparing, you know, policymakers in Washington or just the general public for this new wave of new types of therapies that we've never seen before that are going to look and be manufactured, possibly cost differently to what medicines are now. These are not just aspirin. Patisiran is, well, you know it better than I. If Moderna is successful, their drugs are going to be incredible. How are you approaching those conversations?
Well, one thing that we're doing is we're spending a lot of time with the FDA to make sure that they have a modernized approach to thinking about drug reviews. And we're very fortunate to have Scott Gottlieb as the FDA commissioner. But candidly, you know, a lot of the creative thinking at the FDA started under Peggy Hamburg's leadership. And Peggy, you know, together with efforts on the Hill and also efforts from industry, helped pioneer 21st Century Cures, which is a major piece of legislation that was enacted in late 2016. And that's an example of the type of modernization approaches that have occurred at the FDA, but it's only just continuing. And the FDA is even, you know, as we speak, issuing new guidelines for how to think about gene-based medicines, how to think about, nanotechnologies from a CMC chemistry perspective, but also from a regulatory framework perspective. I mean, it's conceivable in some cases that if you have a real platform, that the review approach for the payload, if you will, if it's a nanomedicine or nanoparticle type technology, can be fundamentally expedited compared to how it used to be viewed in the past, where everything had to be reinvented. And there are, I think, science-based approaches without lowering the bar from a safety and efficacy perspective that can be applied. So I think we're not only seeing this wonderful period of time with innovation coming out of the academy and industry, but we're also seeing a wonderful period of time at a policy level from the FDA around how they will think about the review of those type of medicines and how we can expedite them getting to patients more rapidly without lowering the bar of efficacy and safety.
And Bob, I know that you've spent a fair amount of time in Washington on FDA advisory boards. How have you approached those conversations over the years, advocating for science, making sure basic research is able to get funding in these new areas?
Yeah, well, really the same way that John did. I mean, when I chaired the FDA Science Board, the goal was really to make decisions that were science-based, which might sound obvious, but it's not. I mean, and because sometimes things could be politically based, they could be religious based, and the goal is really to make it science-based. And so to that extent, I mean, what was good about the Science Board is all the heads of each of the divisions would come to those meetings, and we would try to make sure that the most up-to-date scientific advances were brought before them in terms of analyzing data, computer technology, IT, everything to try to, and just making sure that people would have as good a knowledge of science as possible. And I feel that works. I mean, I think that what John said and what I'm saying, I mean, I think that's really what you want to do, is make sure that as long as the science is really good and they're making decisions based on science and not just on opinions or politics, I think that helps, and I think that's really what we've all tried to do.
I'd even think another, if I may, just another area, is on intellectual property, right? And that's another area that's really important here. And because there's been a weakening of the IP system in the U.S. over the years through Supreme Court actions and even some of the legislation around America Invents Act. And I think there is an opportunity to work together to celebrate invention. We're going to have patent number 10 million is about to issue this year. Right. Which is going to be a real landmark for our country, right? For the inventiveness of our country. And we have to make sure that we celebrate the importance of patents, because these type of technologies need a strong intellectual property foundation to be able to get the funding that's needed to bring them forward. And without that, if we don't have a strong system, we won't get the needed capital in place to basically be able to invest in it. So it is important that we all celebrate this important aspect of our society with intellectual property and patents, because it's an important part of bringing these type of medicines forward.
Well, we have about 5 minutes left, and I want to end on this point, that in researching this panel, I was pretty surprised to find that a lot of research, promising research, never does get translated. It remains in the labs. I know that MIT probably does a better job of translating than maybe some other places, but I was really surprised to find that. So I'm curious if, Paula, we can start with you about how you see overcoming that challenge, how you think the academy and the industry could work together moving forward on these really pressing, cutting-edge areas like nanomedicine.
I think starting with what's going on in lab, it's helpful to think about what material systems or devices you're generating and whether or not they are systems that would be translatable in a health or in vivo application. Once you begin to have that mindset that you really think that you can get this across and into the bigger world, you begin to think differently about what you choose to use. Everything from your material sets to the kinds of animal models that you use. How realistic are they? Will they give me a good idea, or at least give me some idea of whether or not my system can meet an immunological challenge or have the correct biodistribution? So getting you and your lab in the mindset of this is going to be real, we need to put this inside a body or deliver this to a patient, changes the way you think about the problem, and it makes you think more meaningfully about what is possible. That and increasing conversations with people who are in industry so that you get a better sense and are grounded.
Does anyone else have any other thoughts on that?
I'm happy to. I mean, I guess the premise of the question assumes that things that are done in research labs should all be translated. And I'm kind of struggling with that premise because I think that most of it shouldn't. I think that we do science because it advances our knowledge, which leads to further questions, would it further advance the knowledge, And at some point, some of that knowledge can be applied to do useful things, but not all of it has to be. Otherwise, we should change what it is that we fund and kind of always work backwards from something that we know can be useful. And that would create, in my view, a pretty big shift. So there has to be, I think, science in support of advancement of the knowledge that is eventually leading to an application and a translation. So I'll maybe say this. So I'm sure there's a lot of science that can be translated that's not being translated, but that's not all of science. I actually think the whole space is a tiny part of what is my view. And I'll say it in a different way. Relatedly, I think that a lot of big innovations that we talk about these days, gene editing has been the most recent one, but just about every one of these things, when they gain popularity or the world pays attention to them, the people that happen to be there at the end end up getting too much of the credit for it and end up getting the story the way they want the story told. But actually, if you then go backwards, and sometimes people do that, you will find traces, evolutionary traces of that eventual innovation, innovation being the translated version, that is absolutely everywhere, and it's that assembly that leads to it. So I actually think that these innovations, what you're calling translation, are often a gradual assembly of things, and so I'm hoping that all of that precedent stuff keeps happening so that the last bit happens, and I also hope that when the last bit happens, people get fair credit for all of the things that went into it, and I often find these days they simply don't.
It's a great lesson for us in the press. Does anyone else have anything else to add here?
So I was just going to say, I think there are, I mean, I think your point, though, is there's something called the valley of death that happens to, that where a lot of things don't get translated. I think part of it is funding. More funding for research is something that we all want. I think John's point about patents is right. I also think, though, that if you're talking about other schools, I think the kinds of things that MIT and Stanford have done have been really good models for what schools can do. I mean, as I've looked at MIT, I should say, as I've looked at some universities that I've been to, sort of the judgment of success is how much money do they bring in the short run. Whereas I think if you look at MIT or Stanford, the goal is impact. And I think impact is really a really good way to look at it. How much impact are you going to make? And when you look at what's happened at Kendall Square or Silicon Valley, I mean, they've made enormous impact. I mean, Hewlett and Packard, if you just take Stanford, I mean, they didn't have any royalties or anything from them, but they certainly gave back plenty to Stanford. And you look at the things that have happened around the MIT area in Kendall Square, and it's just staggering.
And I just wanted to, when Paula was speaking and Bob's point now, it really speaks a little bit to the culture of groups and teams of people that are, and I think MIT and Stanford are just fantastic examples, and I've talked to other universities that emulate what's been done there. I think a lot of it's just the culture of believing in making that impact, right, and ultimately seeing these amazing discoveries get translated all the way through, and that's a cultural thing. Some academic groups may not feel that that's their that's a priority, and that doesn't get done as a result.
Or maybe what may be helpful, at least in my case, what I've experienced is a couple of role models just to show that, you can start up. I agree, not all research is translatable. There is a lot of fundamental research that has to be done, but if there is a chance, if you see somebody who has done it already in the past, you may get inspired. There may be some trainings to get acquainted with IP, with financing, and so on. And if you meet the right people, then it may go into the right direction, and you may go to the valorization path.
To that point, your colleagues are often your friends. I mean, you would go to them and say, what are some critical issues that you think are that need to be addressed in your particular area? And you work with them so you can define something that has some translational potential if that's what your interest is. You can do this as a team, and the team science approach works really well for this. You do it early. You don't wait a couple of years and then go back and say, ooh, I should have asked my colleague this. You just do it early, and I think that it pays off in the end because you evolve together to translate something of interest for the community.
Wonderful. Well, I want to thank all our panelists for being here. Thank you so much for your thoughts.